Hi there, and welcome to a video on enzyme inhibition and the classes of enzymes. So enzyme inhibition refers to preventing an enzyme from working properly, and there are two types. There are competitive inhibitors, and they bind right to the active site, and those are typically going to be toxins in the cell, because they will, a lot of them bind either irreversibly or for such a long period of time that they don't come off of the active site, so that the proper substrate can never bind, or it's prevented from binding so much the process breaks down. The non-competitive type inhibitor is more of a normal metabolic control. So there are times when the enzyme's there, but you don't need it at that particular moment. So for an example, if you're eating a meal, maybe the first 400 calories, are sent down one pathway, but the remaining calories maybe are sent into a storage pathway. So depending on how many calories you're using at that time. So you may need to kind of shut down that without, don't want to destroy the enzyme, but you want to shut it down. So they'll bind somewhere else, but what that does is it changes the shape of the active site. So they both have the same net effect. The competitive inhibitor prevents the substrate from binding because it literally is blocking the site itself. This is your competitive type. The non-competitive type binds elsewhere, but it changes the folding such that the substrate also no longer binds. So same sort of net effect. With the non-competitive inhibitor, it's a little bit easier to remove that and take that off, and then this will go back into the correct conformation that's there. So non-competitive are generally not considered your toxic group. They're just part of your normal enzyme control mechanism. We also can control enzymes by the concentration of the enzyme in the cell. So some cell enzymes are needed at constant levels, and these are called constitutive. So I think of your constituents. If you're a politician, those are your voters that you're kind of relying on to come forward and vote on you. So they're at that constant level, these constitutive enzymes. So normal cell processes, so muscle contraction, DNA synthesis, cell repair, making energy, although there's times actually that's even sent in different levels as well. So they're there at pretty constant levels in the cell. The regulated type are for enzymes that you don't need all the time. So maybe there's enzymes that you're utilizing when the temperature goes up, so that heat response enzymes, or enzymes that you're using as your, maybe extreme exercising. So something is changing in your environmental conditions, so you then there's control in the cell in terms of making or synthesizing the enzyme, and that's at the genetic level actually. And that'll be discussed in other videos, but that control mechanism allows the levels to fluctuate as you need them. So if you're not in a hot environment, you don't need to make the proteins that respond to that. So bacteria, for example, make a set of proteins called heat shock proteins. Some bacteria do. And so when conditions get hot, so maybe you're running a fever, some of these bacteria can actually make special enzymes that help keep them alive a little bit longer. Another example in bacteria would be there are some that make endospores. So that would be a very different set of enzymes needed to make these resting spores. And those would only be made when conditions are extreme, maybe very dry, or there's a certain chemical compound there that is preventing the bacteria from growing. And then finally, there are exoenzymes. Those are enzymes that are secreted outside of the cell, and often those work in digestion and detoxification, because you're trying to break down something that's in the environment that's there. So for example, fungi will break down their surrounding environment in many cases to absorb those nutrients, because they have to take in those nutrients. So if you're going on a hike and you see, in a moist area, you see fungus growing on the bark of the tree, that's actually a digestive process. It's secreting enzymes so that it can slowly kind of attach and digest and absorb nutrients from that tree. Same thing is happening in your spaghetti sauce. If you have mold growing, there's a secretion of enzymes that are there. So that simply just is where they are located. Also, antibiotic resistance is often an exoenzyme process, because the bacterial cell will secrete the enzyme, if it has it, outside of the bacterial cell to break down or detoxify the enzyme before it even comes into the cell to destroy it. And then endoenzymes are what we probably think of most of our enzymes as falling into this category. They are inside of the cell, and they work in all your day-to-day processes. So a lot of those are your constitutive, but some would be regulated as well. So anything from, again, DNA synthesis to manufacturing molecules for the cell membrane to manufacturing digestive molecules, all of those are going to be inside. So I'm using digestion again, but you all also do some digestion inside of the cell. So just to revisit, inhibitors can be competitive or non-competitive. Enzymes can be at a constant level, constitutive, or regulated. And then, depending on where the enzymes are located, that's yet another way to classify them. I thank you very much, and please contact